Bacterium's anatomy is quite simple, being very much like a human or plant cell, throughout defined inclusions, which can contain fats or sugars or other things used by the bacteria. We also find in the middle is a combination of DNA chromosomes that make up the brain of the bacterium. The cell wall of bacterium is typically made of layers of fats and proteins, providing protection to the organism while allowing certain nutrients to get into the body of the bacterium. Within the wall we find the cell membrane, a thinner collection of proteins and fats that allow nutrients in and excrement and secretions out. On the outside of the wall of membrane we find three different types of hair-like filaments. The first are the fimaria, which allow bacteria to attach themselves to surfaces or to each other. These are used to form biofilms. Then we find the pili, singular, pilux. These are tubes that allow the bacteria to connect with each other and exchange information. We might think of this as a form of wire connector, allowing bacteria to exchange things like antibiotic resistance between each other. In other words, when one bacterium figures out how to evade a particular antibiotic, it will then exchange that information with another bacterium. That exchange takes place in the form of what is called a plasmid, which might be compared to one spy exchanging a briefcase with another spy. Then there are the flagella. This diagram shows a single, but as we discussed earlier, there could be many of these swimming appendages of sorts surrounding bacteria. We mentioned earlier that bacteria are often classified by whether they are gram-negative or gram-positive. What's this? The terminology comes from the laboratory from what is called a gram stain. Some bacteria can be effectively stained while others cannot. The difference lies in the cell wall and membrane of the bacteria. The gram-negative bacterium will have a much more complex and hardier outer cell membrane made of porin proteins with somewhat of a double wall. These resist staining more than the gram-positive bacteria. Gram-positive bacteria do have a pretty thick membrane as well, but the membrane is made up of the more permeable and more regular proteoglycans. These will let in the staining and keep the stain, allowing it to be visible. Outside of staining, the main differences relate to the lipid content of the gram-negative bacteria and the fact that gram-negative bacteria can readily transfer plasmids, which are antibiotic-resistant information, to other bacteria. This means gram-negative bacteria are more likely to become antibiotic resistant.